We're going to, keeping with the multidisciplinary activities of the BDP, we're going to change the topic a little bit here and talk about something that's occurring right now and has been occurring with increasing frequency in our world, and this is the concept of emerging infectious disease. There's 1,407 organisms encompassing fungi, bacteria, parasites, protozoa, and viruses that are capable of infecting humans and causing significant disease. 58% of these are zoonotic. In other words, they're animal diseases that have jumped from animals to man. And this process has been going on since the first human colonies of mankind. Our measles virus came from dog distemper. 177 of these 1,407 organisms are considered to be newly emerging or re-emerging diseases. If you've been watching the news, there's an outbreak of bubonic plague in China at the moment. They've had one death. And again, the majority of these new pathogens are animal diseases that we've either never seen before, haven't recognized before, or they're old things that we thought were conquered and have now reappeared. During the past 30 years, we've had 41 previously new and completely unrecognized diseases appear. And this risk continues to grow globally for a variety of reasons. This is a very partial list, starting with the HIV/AIDS virus back in 1908. This has been molecularly traced back to a small area of the Cameroons to chimpanzees. In the 70s, something significant happened to our country with the inadvertent importation of Aedes albopictus, the Asian tiger mosquito. It came in through some tire casings in Houston, and it's colonized our country. This is the vector, insect vector, capable of transmitting a variety of things like dengue, West Nile fever, and some other pathogens. In 2013 to 2014, we've had a number of outbreaks that we're concerned about. One is the Mideast Respiratory Syndrome virus, a new coronavirus in Saudi Arabia and the United Arab Emirates. This looks like it was imported through camels from Australia and that it was originally a bat virus. It's carrying a very high death rate. The Basque Congo hemorrhagic fever virus, this is the first time we've seen a rabies virus or a rabies type of virus from the same family capable of causing a hemorrhagic fever. The current outbreak of Ebola in Sierra Leone and Liberia, that seems to be expanding. We'll touch on that a bit. The ongoing epidemic in the Kasai Oriental province of the monkeypox virus. This is a relative of the smallpox virus. It does infect humans, and it appears to be changing in its aggressiveness. A few weeks ago, we got our first cases of chikungunya virus in Florida. And the second case was transmitted by mosquito. So it's in the population of mosquitoes there. And like the West Nile virus, which is now virtually all over the United States since 1999, we expect this to get worse. And then we've had 2 cases of dengue virus in Florida. So this is an ongoing problem. It's becoming worse and worse. And is it linked to climate or not? I think the main link is between our ever-expanding growth of our human population. Never before in human history have we lived under these population densities. And without very strong continuous public health, which we're not seeing, this becomes an increasing risk for all of us. These pathogenic zoonotic or animal-borne viruses don't exist in a vacuum. They have their own particular dynamics in the wild animal population that serve as the virus reservoirs. Most particularly worrying are the RNA viruses, because these infect a human cell or an animal cell. They require that cell's machinery to replicate. And when they replicate, they tend to make a mistake. Their enzyme called RNA polymerase isn't very accurate. And within quite short periods of time, you can see these viruses start to change into different clades. The MERS virus has only been out for about 2.5 years now. We've already seen it develop into a B subclade population after only about probably 800 total cases. So these are a constant worry. And then on this Venn diagram, you can see this intersection between human populations, the wild animal reservoirs, and the transmissibility of these RNA viruses. It very often leads to nothing. Occasionally, it can lead to unrecognized human diseases that may take decades to become apparent, such as HIV, the AIDS virus. They may not, at the moment, cause human disease, and we're very worried about something called the simian foamy virus in Africa. Or it can turn into something quite dramatic, and something like the Ebola virus. Viruses can affect different species differently. This is an example of the tropism of the Rift Valley fever virus. In lambs and horses and humans and monkeys, it can cause very severe disease. Guinea pigs are completely immune. For a virus to cause a human infection, we go back to what we call the lock and key theory. The virus has to have a protein on its cell surface, which is the lock, and it looks for a protein receptor on its target cell, which is us. And if the lock doesn't fit, the virus can't enter the cell, and it can't cause an infection. So, there's a very species specificity to a lot of these RNA viruses. The problem is it can change species specificity. These animal reservoirs of human disease, there's a variety of factors involved. Is this organism transmitted by an insect vector? Well, then we have the population dynamics of the insects. Now, that may be susceptible to global climate changes. The lifespan of the predator, what's its food supply, what's its range, does it migrate? We have a problem with the influenza virus every year because of the migratory birds. There's one crazy bird, the Arctic tern, that flies from the north to the South Pole. And when it gets done, it flies back to the other pole. This is how these isolated Eskimo populations ended up with the 1918 flu virus, through the bird droppings. So this becomes an important factor in these things. The infectivity. How much virus does it take to initiate a human infection? Very often, animals can be very susceptible to a small infective dose, whereas humans require a much larger loading dose. In this type of case, we have an intermediate animal that usually amplifies this. And then immune or non-immune populations. We just mentioned this amplifier host. One of the good examples of this is the Hendra virus. We found out about this in the mid-'80s. And it was an unusual outbreak of very, very severe lethal disease in Australia, and it occurred among some horse handlers. What they found was there was an infected bat colony near human habitation. And these horses, they were fruit bats, and they like orchards. And there was a fruit tree, and these bats had congregated underneath it for several days. And their droppings had gone down, and this was one of the few areas of shade on this farm. The horses were congregating underneath it, and the horses got sick. They picked it up from the environment at low dose. The virus amplified in their bodies to tremendous quantities, and the veterinarians got ill and the horse handlers. So this is how secondary human infections occur. The Nipah virus in Malaysia in the 90s. Again, infected fruit bats. Virus was shed into the environment, and in this case, the amplifying host was domestic pigs. So the first illnesses we saw, these pigs had a low threshold for infection. Only a few viral particles would infect them, but they were able to secrete large amounts of virus, and the first human infections we saw were in the abattoir workers and the people that handled the animals. So these emerging human infections usually go through a couple of phases. An infectious organism will try to expand outside of its initially well-defined geographical area using usually a single species of animal. If this becomes successful and it ranges out of its geographical area or becomes endemic to a particular region, it'll try to expand its host range. And this is just done statistically by random mutations. As it's able to expand other types of animals, this now becomes more serious. And RNA viruses sometimes tend to shuffle genes around between other RNA viruses. This happens with the influenza virus. Every year, its genetic material is divided into 5 components. So you're infected with a bird influenza, at the same time you're infected with a pig influenza, and the genetic components shuffle, and you end up with a new type of human influenza. This happens every year. We try to guess what is going to be because our vaccine production methods for this are out of date and the FDA won't increase it. But we try to guess what strain's going to become pandemic, and that's in your flu shot. Sometimes we get it right, sometimes we don't. So once this starts, this is all, everything is setting up for something to undergo an outbreak. So our species numbers has doubled in the last 27 years. This is an ever-expanding global population. At the same time, we're fragmenting or destroying a variety of these natural habitats. Their food sources for these animals become less diverse. And then we compound this with our intensive agricultural process, this battery raising of chickens, these massive stockyards. And these provide ideal environment for viral mixing and genetic material exchange with their wild cousins. Urban parks, landscaping nature reserves, global temperature-induced insect vector animal reservoir migrations. And again, the biggest risks come from these RNA viruses. I almost hit a deer in downtown Washington, D.C. a couple years ago, a white-tailed deer. So that is not its natural habitat. And the ticks it carries, probably infected with Lyme disease. And we're seeing Lyme disease expand continuously throughout the United States as time progresses. The hemorrhagic fever viruses are a unique type of infection. It actually encompasses quite a number of different viruses. They all start with about a 1 to 2 week prodromal period where you just don't feel well. You feel like you have the flu. You run a high fever, you have muscle pain, gastrointestinal disturbances. And then depending on really what virus you're infected with, your clinical course changes dramatically. For the Machupo and Lassa fever viruses, you get these ulcerations in the oropharynx. Crimean Congo fever, your platelet counts drop dramatically and you start bleeding into the skin and mucous membranes. The well-known yellow fever, as well as dengue and Rift Valley fever, attack the liver. It can give you a jaundice syndrome. Dengue is a particularly odd organism. There's 4 serotypes. If you have one of those serotypes, an infection, this is the infection where you're afraid that you won't die. It's very painful. Its old name is breakbone fever. It used to be endemic in Baltimore. during the founding of our country. That's fine. If you get infected with most people recover. Children carry the most mortality. But if you're infected with a second dengue serotype, your immune system makes a mistake and you start to attack your own platelets, and now it becomes hemorrhagic. The Hantan virus, both the Korean hemorrhagic fevers that we first learned about in the 50s, and then the Four Corners agent in the 90s that broke out west that caused a pulmonary syndrome. And then the well-known Ebola and Marburg viruses. And these viruses are very typical because they infect a lot of different cell types in your body and set up a multi-organ dysfunction syndrome. So we have the Bunya viridae. This is Rift Valley fever, Crimean Congo hemorrhagic fever. This is a Caucasian patient in South Africa with Crimean Congo hemorrhagic fever. The platelets counts are almost nothing. You can see this massive ecchymosis of bleeding into the skin, subcutaneous bleeding. We have the arena viruses like Lassa fever, Machupo, Argentinian hemorrhagic fever. The flaviviruses, as we mentioned, yellow fever and dengue. And then the filoviruses, Ebola and Marburg. The Bolivian hemorrhagic fever is interesting because it just summarizes everything we've just told you about emerging infectious disease. This broke out in the 60s. It's a virus with an incubation period of 2 to 21 days. The victims develop a high fever, headache, nonspecific muscle pain, diarrhea, nausea, conjunctivitis, very extreme. And then they progress into these small bleeding spots, mucous membrane bleeding, and bleeding into the skin. And followed by jaundice, major organ necrosis, renal failure, encephalitis, shock, and death. And it carries a very high mortality rate. Well, this was actually socially inspired, this outbreak. Back in the 50s, this isolated village of San Yanquin in Bolivia had a population of 2,000 people. Huge tracts of rainforest were cut down to provide grazing for cattle. And some Brazilian magnates had moved in, bought up all the land, and they were raising cattle there. And the whole purpose of this town was to support this cattle production. The town, there was nothing around it. It was totally dependent on this company steamship that would come in once a week and supply everyday commodities to the it's essentially a commune. And everything went perfectly well until 1952, when a political revolution brought in the Movimento Nacionalista Revolucionarios to power, and their platform was a policy based on land reform. So they threw the Brazilian magnates out. Well, the steamships stopped coming. No food, so the population immediately started developing crop fields for corn. This was quickly cultivated and irrigated, and corn in this area is the staple food. Well, with all the irrigation and stuff, malaria became a problem. So they brought in some organophosphate pesticides, and these inhibit the neuromuscular transmission, the enzyme acetylcholinesterase. And a lot of the cats died. In 1962, then there was this sudden mysterious disease that broke out. It affected quickly over about 50% of the inhabitants over the next 14 months, and the villagers started calling it the black typhus. Small U.S. combined with Bolivian investigation team arrived. This was Carl Johnson at the CDC at the time. And they formulated a case definition. This is where you come in and define what is going on with this disease. You list its important characteristics, and you try to search for a cause in other cases based on these. And they found it had a 7-day incubation period. This is an example of this massive ecchymosis bleeding into the skin of one of the villagers. And death could occur within 4 days of symptoms. and that there were two patterns of disease. One was neurological, characterized by loss of balance, grand mal epileptic seizures, and a hemorrhagic meningitis around the brain. And then a hemorrhagic disease with this mucocutaneous bleeding, eventual development of shock and death. They mapped all the cases in the village, where they lived, what they possibly could have in common. They interviewed all the families and relatives and neighbors, and the only thing they could find was the risk for catching this disease was living in this village. There was no clear transmission route that could be discerned. Carl Johnson came back with a small laboratory, and again, this is an isolated area, so you're really doing frontier science here. They set up animal traps, and they set up a small diagnostic laboratory. And I always call it the diagnostic dog. When you don't know what's happening with a patient, inject a dog with its blood. Anyway, it's more sophisticated than that. And these are inbred laboratory animals, so we can get reproducible results. The breakthrough finally came when they were allowed to autopsy a 2-year-old boy who died. And they ground up portions of his spleen and brain, and they injected it into these on-site experimental animals who promptly developed the disease. They didn't have electron microscopes out there, but we've known how to determine this stuff for a couple centuries now. And they used filters to determine that it was probably a virus causing this. And then they did widespread biomedical sampling. And they found these sick gray field mice in the traps. And they were able to show with their experimental animal system that the virus was present in the rodent's urine, and this was how the disease was transmitted. This was the local host of the virus. Well, what had happened when the village started growing massive amounts of corn and the organophosphate pesticides had killed off the cat population, there was no natural control. You upset the ecological balance, and the mouse population exploded. So the village was overrun with mice. They would come in at night, run over the corn, stored corn, run over the tortillas, whatever. And eventually it affected 20% of these local inhabitants. Mouse urine would contaminate the foodstuffs or contaminate the dust. And as you walked, you kicked it up and you received an aerosol inhalation, like the Four Corners agent in the 90s. Or you could have direct contact from picking up and handling the dead mice or touching infected human patients without gloves, coming in contact with their body fluids. In fact, Carl Johnson himself got ill and almost died. Normally we try to name these things after the nearest river. In this case, it was called Machupo. They initiated a program of rodent trapping, and they brought back in the cats, and the epidemic was over. So this is a very perfect example of how emerging diseases occur. There's an upset in this delicate, violent. Man comes in contact with these things that normally wouldn't be a problem, and you have a new disease. We're going to talk a little bit about the filoviruses, because everyone's heard of Ebola, I assume. And there's a raging epidemic going on now in about 4 countries in Western Africa. In 1967, there were 31 cases of this unusual hemorrhagic disease that appeared simultaneously in a vaccine production factory in Marburg, Germany, and Frankfurt, and in Belgrade in Yugoslavia. And they were making viral vaccines using monkey kidney cells in these bioreactors that you see at the bottom. And all of these workers had had contact with the blood organs or cell cultures derived from a batch of green monkeys that had been imported from northwestern Uganda. It was an unknown hemorrhagic fever. Nobody had ever seen anything like this before. 7 to 10 day incubation period. Sudden onset of back pain, sore throat, muscle pain, headache, nausea, the normal prodromal signs that your immune system is making a massive response, followed by an unusual maculopapular skin rash, headache, increasing fever, thrombocytopenia, and bleeding into the subcutaneous tissues and the mucous membranes, lining the mouth and the gastrointestinal tract. These patients rapidly progressed into a hepatitis with jaundice. Once the liver becomes involved, you lose your vitamin K-dependent clotting factors, so your bleeding conditions worsened even more. and they died from encephalitis, hepatitis, pulmonary and gastrointestinal hemorrhages. Samples were sent to Fred Murphy at the CDC, and this is very often how we discover things, with direct visualization. They examined it under transmission electron microscopy, and here was this previously unknown viral structure. They called them a filovirus, after the word thread, and they named it Marburg, after the town where the sample came from. This is an immunohistochemistry in a cell culture, and everywhere you see red is live Marburg virus, genetic material. 6 secondary cases occurred. 5 of these were in healthcare workers who'd had contact with the body fluids or blood of the primary cases. And this is a story that repeats again and again and again and again. One secondary case was the wife of a patient who became ill 83 days. He recovered. They sent him home, and his wife came down with it 83 days later, and he was excreting active Marburg virus in his body fluids. In this case, the virus damaged the lining of the blood vessels. Your microcirculation, your tiny, tiny capillaries. There's not one cell in your body more than 1 cell diameter away from a capillary. And really all we are is this capillary meshwork with our cells put on top of it. And if that capillary bed becomes damaged, it can be irreversibly damaged, as you can see in these diagrams. A process of acute inflammation, and then you trigger your clotting system, you get microthrombi forming, your white blood cells become stagnant, they release more inflammatory chemicals, and you proceed into multiple organ dysfunction and death. The patients that do survive have a very prolonged convalescence, permanent or temporary hair loss, it does grow back, furrowed nail, because the nail beds have been damaged, permanent deafness, uveitis or encephalitis. And when we went back and started to look in Africa, we found that these things were occurring all the time. Second outbreak occurred in the country in Rhodesia. She died in Durban. An Australian hitchhiker picked it up in '75, passed through Rhodesia, died in South Africa. French plantation workers, Kenya, 18-year-old male in '82, and now the country is Zimbabwe. On and on and on and on, and we find that this thing is endemic in parts of Africa. In fact, if you go back, I'm a great fan of Albert Schweitzer, and if you read some of his old books, you can find descriptions of what looks like Marburg disease way back then. In 2007, there was an outbreak in Uganda among the lead miners in southwest Uganda. CDC, South Africa, Bob Swanepoel is the lead investigator there, and WHO scientists went down there, and they were starting to focus on bats before this time. Fred Murphy was fairly convinced that this was where it was coming from. And they found a nearby cave called Kitaka Cave. They went inside with protective gear, and they found 100,000 Egyptian fruit bats, and they managed to sample 800 of these bats. And they came back in a second expedition and examined over an additional 1,000 bats. And they color tagged these and released them after drawing the blood. Bats don't like collars on their legs, so they put it around their necks. 13 of these bats had antibodies to the Marburg virus. 31 bats had actual Marburg RNA fragments inside in their blood. And they got live Marburg virus from 5 samples, demonstrating that this bats were probably the natural reservoir for this. They looked at it genetically, and it looks like this has had an extended history over the last several thousand years, and it evolved within this species. And in the end story, about 5,000 Marburg-infected bats flew out of this Kitaka Cave each night in search of fruit trees or gardens. In 2008 in Uganda, a 41-year-old female tourist from the Netherlands visits Python Cave in a nearby forest. This was 30 miles west of the Kitaka Cave. She made it back to Amsterdam and 13 days later became ill with pyrexia, headache, maculopapular rash, conjunctivitis, bleeding, organ failure, and death, and she was diagnosed with Marburg. South African and CDC scientists roared in there to this cave 2 months later, and they found one of the tagged bat collars from the Titaka cave, indicating that some bat came out to visit its friends, and that's how these outbreaks are occurring. Well, it still doesn't explain where are the bats getting the Marburg virus from. When we look at this virus genetically, it's only about 2,000 years old. So is it endogenous within the species? Is there an insect reservoir? Well, these things don't really eat insects. They eat plants. So is there a plant reservoir? Why is there so much drama about a disease that's killed less than 3,000 people over a period of the last 40 years? There's movies about Marburg and Ebola and all this stuff. The reason being was during the old Cold War, and possibly still today, the Soviet Union weaponized Marburg as a dry agent. And next to smallpox, this was the most effective biological warfare agent on the planet. They grew it up in guinea pigs rather than tissue culture. And the guinea pigs would come down on an assembly line and be injected, and then 4 days later harvested for the virus. And this process continued until one of the researchers named Ustinov accidentally injected himself. And he died after 14 days, and that became their weaponized strain because it had passed through a human. And the Russians still won't give us a sample of that to look at. Anyway, they loaded this on intercontinental and intermediate range ballistic missiles. The Soviets tended to use everything dual purpose. These are the canisters. They could be used to carry chemical warfare agents or 3 biological bomblets. This is the Russian design. And the agent fill is here in a small explosive burster charge. These things would spin as they were dropped from an aircraft or a missile warhead, diffuse out laterally, and detonate when they hit the ground. They would generate a small particle aerosol in the 1 to 5 micron size range, which would be taken into the lungs and retained to cause infection. Back in the 90s, the Soviet Union had collapsed, and one of our agencies was able to get a small team into their testing site on the Aral Sea, Renaissance Island, to take soil samples to try to find some of this stuff, but it didn't work too well. In '76, a new filovirus emerged in Africa. It broke out simultaneously in the Sudan among cotton factory workers and at a Belgian missionary hospital in Yambuku, Zaire. The outbreak was termed green monkey fever. Tissue samples were sent to the CDC in Atlanta, and they found this brand new filovirus. They named it after the nearby Ebola River. If you're interested in this stuff, Glenn Close, the actress, her father was the doctor at Yambuku Hospital, and he wrote a nice book called Ebola, which is interesting and describes the first outbreak of this. Looking at the genomics of Ebola and Marburg, they're very, very similar, and their rate of genetic divergence is about the same as the hepatitis B virus. Tracking backwards with molecular clocks, it looks like Ebola and Marburg originated from a common viral ancestor about 2,000 years ago. Ebola there's only a few amino acid changes in its outer protein envelope or protein structure, and this gives a much different disease on the biochemical level. Incubation period, 2 to 21 days. An acute illness, 7 to 10 days. Again, you blow this very fine maculopapular rash. Healthcare worker infections seem to have a higher mortality than the primary cases. And if you live through it, you have a very prolonged convalescence. With Ebola, the liver involvement is much, much, much less than in Marburg virus. Acute onset of high fever, Malaysia, general malaise. This rash that develops on the face and chest on day 2 to 3 of infection. And then this your body starts making enormous amounts of tissue factor. This triggers your clotting system, and at the same time your platelets start to drop and you start mucous membrane bleeding. Like the HIV virus, curiously enough, there's a large drop in your circulating lymphocyte count of your CD4 and CD8 lymphocytes, your helper lymphocytes. So not only is this virus replicating and taking over, but it's destroying your immune cells that are responsible for containing the infection. A lot of your tissue enzymes are released into the bloodstream from the damaged cells, and then you start this disseminated intravascular coagulation, and you're in the agonal phase with an encephalitis. The virus is easily diagnosed. These are a list of the methods. You can grow it up very easily in cell lines. There's RT-PCR reactions for this. Management's difficult. We do seem to have a tentative vaccine now based on a vesicular stomatitis construct, but it's basically supportive therapy. There really is no cure. One thing that provided a longer survival rate in monkeys was the nematode anticoagulant protein. This is what the leeches secrete into you when they latch onto you. That provided several days longer survival in monkeys. Working with Dennis Kleinman at the FDA at the time, we found that small little synthetic fragments of DNA, heavily methylated, seemed to amp up your immune system. And it provided a role that your own immune system was playing a big role in the patient's death. A superactive immune response was helping to damage the tissues. And we found that guinea pigs, we used a guinea pig adapted Ebola, and we found that if we could inject these synthetic DNA fragments either before or within 24 hours after injecting them with Ebola, that their fur didn't even ruffle. They didn't get an active infection. So I lost interest in it. It didn't work for some of the other viruses. Your risk for infection, this is street meat. Protein is very scarce in many parts of Africa. And rats, monkeys, this type of thing are sold in the markets. Attending the funerals of Ebola victims, coming in contact with their family in Africa, providing care to an Ebola patient without wearing protective equipment, laboratory accidents, or going into caves in Africa. These are all risk factors. And it's not the dreaded disease that it's made out to be in the popular press. If you'll wear a mask and gloves and decontaminate yourself with bleach when you're done examining the patient, you don't need a sophisticated facility to nurse these patients. Just basic things we've known for 2 centuries. 1989, there was an Ebola outbreak in Reston, Virginia, at a primate holding facility, the Hazelden Primate Facility. There's a picture of it above. Some samples ended up at Fort Detrick. You guys, because Fort Detrick handles a lot of monkeys and they have vets there, they put it under the electron microscope and had a heart attack. It was an Ebola virus. So they came down and completely decontaminated the quarantine facility. Several workers tested, later showed antibodies to Ebola. They'd been exposed, but nobody so much as run a cold. Well, this was very confusing. They were found that these monkeys were co-housed with monkeys from the Philippines during their transport to the U.S. And a second outbreak occurred in 1996 in Texas among animals from the same Philippine supplier. So obviously it's time to go over and look in the Philippines. Only one paper came out of that. But the Ebola Reston virus, by its ability to kill monkeys and by its lack of an ability to cause death in humans, provided some very important clues onto how it was causing human death in the infectious Ebola Zaire strains. Even though there were only a few amino acid differences, we were able to show in tissue culture that it caused the lining of your blood vessels to commit suicide, to undergo a process called apoptosis. And without your microvascular blood supply, your cells went necrotic and died. And this triggered the massive necrosis of your organs and the massive blood coagulation of abnormalities. And this was later confirmed in 2000 by Yang et al. 2007, a new Ebola strain emerged in Uganda. And this lasted for 2 months, 149 suspected cases, 37 deaths. This picture was taken at the Bombo Military Hospital. This is an Ebola patient. And you can see no gloves. They ran out. And that's how these things get propagated. In 2009, Ebola arrested again, and this time the outbreak was in the Philippines, and it was recognized. It was detected in tissue samples taken from pigs suffering from an acute respiratory infection, and it showed that they were infected with widely infected Reston strains, showing it had been in the population for some time, even before it was discovered and had the outbreak in '89. Again, there were no human cases. 5 out of 77 people who'd come in contact with the pigs showed antibodies. But there was a concern that the virus could be an RNA virus. It could further mutate into something pathogenic for man. So we're keeping a close watch on this. This was an example of a failed emergence into humans. There was an infected bat colony, pretty much determined near the abattoirs. Amplifier host was the pigs. They secreted large amounts of the virus, but it was non-pathogenic for humans. In Africa, it looks like the bats are spreading this slowly eastward. It says west, but it's eastward this way. Over a 5-year time period, researchers have found 47 dead animals in Gabon. 7 of these are determined to be infected with Ebola Zaire. The mountain gorillas, the Dian Fossey mountain gorillas, are rapidly becoming extinct from Ebola at this time. And what's a little worrying is we're starting to get these genetic diversions. We're now up to 3 clades of this virus. And then finally, they managed to get a hold of these cute little fruit bats, and they were able to show that these are the animal reservoir. So bats seem to be very important. From 1994 to 98, 4 other new filoviruses have emerged, including the Hendra and the Nipah virus. And something is happening to the fruit bat ecology. These are probably the largest diverse species of mammal on the planet. There's 1,116 species. They're very sociable. They roost in very high numbers. They can fly dozens of miles each night. They do migrate slightly between summer and winter. And we really don't understand a lot about their antibody resistance. Some of these have a lifespan of 25 years. So what's happened is, again, human intervention, drastic land clearing, cattle ranching, urban sprawl, diminished old forest. The bats just find it easier to live closer to human populations. They can gather in large aggregates, travel shorter distances to feed, and their reinfection of their colony occurs less often. When infection does occur, there's a massive die-off of bats. Large amounts of virus are secreted into the environment. People come in contact with this, and then the cycle renews. 2012, a mysterious disease broke out among captive boa constrictors in the United States. This was a neurological disease causing them to twist themselves into knots and to stop feeding. They pulled out a new virus out of here, and they found one of the Ebola genes inside it. So there's a possibility the progenitor Ebola Marburg virus may have gone back to the Cretaceous. It may have been around us for a long, long period of time. In 2012, we discovered a new filovirus in Spain inside the bats. And they're still researching that. Then there's a curious question of Borneo. Survey done a couple years ago for H5N1 bird flu by the Indonesians. They published a paper. They had collected blood from 353 orangutans, and they found 18.4% had various strains of Ebola virus And these were African strains from Zaire, from Sudan, from the Ivory Coast, and from Uganda. 1.7% were seropositive for Marburg virus, and only 5 had antibodies to the Philippines Ebola Reston virus. This was very confusing. Is there multiple species and coevolution going on? Was it inaccurate laboratory testing? Was it differences in the way the tests worked inside orangutans? One thing's for certain, it's too far for a bat to fly from Africa to Borneo. And if Ebola Reston, which is non-pathogenic for humans, is in the Philippines, then Marburg should be there as well. There's not been any recorded Marburg outbreaks there. So we went over to have a look, and we didn't find, it looks like now the paper's going to be withdrawn, and it was bad or inaccurate laboratory work. But what I did find was absolutely alarming. In British North Borneo, this is the Kanapatang corridor, and the government very wisely set aside a 100-mile wide corridor where no development can occur. That's for wildlife. But the palm oil plantations on this side of the river have come right up to the jungle. This is an offloading station for palm oil for the ships that go up and down this large river. And you've got human encroachment right up to the forest. And to me, this is just asking for trouble. It's also taken all the animals in North Borneo and crammed them into this 100-mile wide corridor. When we were over there, I never saw so many monkeys of different species crammed together, living together, to the point where this is my tree, not yours, and this is our tree, not yours. So it's really setting up everything we know about emerging disease. It's setting up for something bad to happen. We have some new tools. These are the DNA chip detectors. The same way that we make microcircuits by lithography, we can etch tiny little channels in these silicone wafers and put little bits of DNA on there. And I've been working closely with Tesserae, one of the manufacturers of these, and we can scan for several hundred viruses at a time. And it only takes one operator, and it only takes 24 hours. So a great, we okay? So a great deal of laboratory can be done by only a few investigators. The goal of all this is called viral forecasting. And there is a global viral forecasting organization. The idea is to recognize it in the jungle before it gets here. And they're only interested in working in Africa. They don't want to work where I want to go. So for the last 5 years, we've been setting up an infrastructure to conduct this type of research. We have a jungle school now in Puerto Rico where we can train scientists. I'm a fellow of the Explorers Club, a national fellow, so I'm working with some other people to try to get an expedition back to this area within a year. And then the long-term goal is to build a floating laboratory to take into these areas. Larry Bell, who's a member of Doctors for Disaster Preparedness, is the Dean of the School of Space Architecture in Houston, and he's been very graciously working with me, trying to get my crude diagrams down into something practical. We're nearing finishing the design of this boat, which I intend to build, and hopefully within the next 5 years we can get this over to the Kanapatang corridor and start doing some research. This vessel's designed not just for biology, but we can bring some microclimatologists and tropical ecologists over and really start, because this will change. We'll try to document how this changes over the next 15 to 20 years. That's the eventual goal of this. Now, for real detailed, using high concentrations of virus, you need specialized laboratories. You can take your samples fairly safely over there. You can do the GNA chip analysis fairly safely. But if you're going to do nitty-gritty work, you need to work in these special biosafety level 4 laboratories. There's a handful of these in the world. Some have hood lines inside, but your main protection are these ventilated suits. You enter through an airlock, and the whole facility is under negative pressure, your laboratory, so nothing can leak out. Most importantly, up until the last budget cut, when they slashed the Department of Defense, we had this aeromedical transport team that could use these isolators in the background to fly anywhere in the world, pick you up if you were sick, and fly you back to Fort Detrick. You would be airlocked in, and you go into the slammer, where it's an intensive care unit, but everybody wears these suits. Well, that's now no longer, as of last year, that's over. Got cut with the budget. If you have any interest in this subject, I would recommend this book. It's well written, it's easy to understand. It's by David Quammen. It's called Spillover. It's in paperback now, so you don't have to pay a fortune. And it's a very good read. Any questions? It's a hood, and it fits inside the poly bag, and you carry, just like the movies, except you look like the Michelin Man, because it takes filtered air and blows it into your suit so you puff out. So you're under a slight positive pressure here, and your airway is under a slight positive pressure, and you wear it for 4 days. But even then, that's like, you really need these laboratories to work safely. So the idea would be to, if we can get this Asian global viral forecasting thing up and running, to make partnerships with the CDC BSL-4 laboratories, if they're still going to be around after this last disaster, or with Fort Detrick, or with the people in Amsterdam. There's a number of these. The French have a very, in Lyon, they have a very, very good biosafety level 4 laboratory. And they're already in Africa looking for these things. So that's sort of the plan. Find these things, send the samples to these high designated security laboratories, and then let them do the nitty-gritty work. And it's not just the filoviruses. We're looking for something that's jumping into the monkeys. And if you go to the surrounding towns, when we were there are monkeys everywhere. They're in the hotels trying to break in and nick food and this and that. And this is the danger, because the monkeys into the small towns, people in the small towns, they don't like them. Somebody grabs one and gets bitten or scratched, and then the game starts. So we're hoping that this would serve as a focus for a variety of different researchers from a variety of places around the world to come. The boat would provide them not only a laboratory, rather than take samples back, why don't we take the laboratory to the jungle? That's the idea behind this. And let the boat provides a place where they can rest, and then go into the jungle and do their work. Yes, sir?
How effective are various agents, like for general use, betadine or bleach,
kill all of them. Yeah, it kills all of it. It kills all of them. These are envelopes, so they're very common detergents kill them. To make doubly sure, we use a substance called Envirochem. Any glutaraldehyde-based product, phenol-based product would work. They're fairly easily killed.
And that viruses in general? Or just
the envelope viruses. Things like the norovirus is almost impossible to kill. The one that causes the diarrhea on the cruise ships, that you have to use a phenol-based disinfectant for that. It doesn't have an envelope. So all the alcohol-based hand washing, it's not, you're just making somebody money that sells it. Sir?
With the border situation that we have in Infectious,
it's horrendous.
what worries are the, of what you talked about, are endemic in, say, Guatemala, Honduras?
What I'm most worried about, and one of the reasons that you have to have some type of border control, is mainly for tuberculosis. Now, it used to be you went and got a chest x-ray. Other countries still require that. We're seeing a resurgence or insurgence of multi-drug resistant tuberculosis to the point where we have extreme multi-drug resistant tuberculosis where none of the drugs are working. And these come out of less than developing world countries. They don't have the health infrastructure. To treat TB is a laborious process, and it requires very strict patient compliance. You can't just stop the treatment when you feel better. And we're getting some strains now with death within a year from diagnosis, and there's nothing we can do. The drugs aren't working, and we only have 2 drugs in the pipeline. And it's got to be researched. So this is a huge worry, and one of the things we want to look for on the boat, we found out now where everything lives. It's an endosymbiotes inside the plant.
Use this for the moment.
It's an endosymbiotes inside the plant. So one of the things we're going to look with at the boat are for some new tuberculosis drugs. Does that answer your question?
I've lived in Florida for 12 years now, and I guess my county stopped the mosquito spraying program about 3 years ago due to economics. But this year, the first year that I've gone outside during the daytime and been bitten by mosquitoes.
Those are the albopictus. They're day biters.
Yeah, and
most of them like to do it at sundown or early morning.
Right.
But the Asian tigers will bite you during the day.
That's the first time I've seen it in Tampa, in the Tampa area. So this year.
We were down there messing around the big lake down there, Okeechobee, and these things are as big as my fingernail.
So what other mosquito-borne diseases, are they separated between night and day?
No, there's about 25 that they can carry. Most particularly, we're worried about the encephalitis viruses. Venezuelan and equine encephalitis. This is the one that closes down Disney World from time to time. We're worried about that. We get outbreaks, huge, 100,000, 200,000 cases in South America with every outbreak there. So we're worried about that. We monitor the horse populations. You know, there's a big thoroughbred horse population in Florida. And these tend to be monitored for antibody levels. And we put chickens out as sentinel animals because the viruses will feed on the chickens.
We're seeing a huge influx of Central Americans too now. Could they be carriers of some of these? Yeah.
The problem with the chikungunya outbreak was CDC quickly traced back the first case, and they were in the incubation period when they arrived. The second case was bitten by a local mosquito in the southern Florida area, which means the virus has gotten into the mosquitoes there. So we expect this to further spread. Thank you.